sharing some preliminary data from an ongoing EEG study that's happening in the aphasia lab and the speech neuroscience lab here at USC. And so I'll just start by discussing sensorimotor integration, what it is, and how models of speech production have approached including sensory information in those models. I'll also talk about sensorimotor impairment in aphasia. Then I'll go on to discuss the goals of this project, what we did, what we found so far, and some suggestions for future directions. So, sensorimotor integration is the mechanism by which sensory information can be used to guide action. It's very important for motor control and particularly interesting to us is speech production. It defines the target of the action. It also provides feedback during planning and execution of the speech act. And for speech, auditory and somatosensory information is particularly important. And a lot of people have examined this in a variety of ways over the years. When prelingual children are deprived of acoustic information, speech and language may not develop normally. And also when adults are deafened later in life, we see maintenance of intelligibility, but we often see declines in articulatory precision, management of loudness, as well as phonetic contrasts. And then with delayed auditory feedback, this has been examined in both normal controls as well as people who stutter. And in normal controls, we see an increase in disfluencies when they're provided with delayed feedback, but in individuals who stutter, we actually see that decrease. And then I'll talk more about altered auditory feedback a little bit later, but given altered feedback, normal controls tend to compensate in the opposite direction of the shift. So in terms of the speech models, sensory information has been included in speech models since the 60s, and more recently, integrative models have argued that auditory and motor phonological systems co-activate to generate a forward predictive model. These models make predictions about the anticipated movements of the articulators, as well as the expected sensory consequences of those movements. So any deviations from this internal model are flagged as an error and corrected. So here we have just two schematics of commonly cited models. On the left we have directions into velocities of articulators, or the DIVA model. This model unifies neuroimaging data with methods for computational modeling, and it includes both feedback and feed-forward control mechanisms that integrate while speaking. On the right, you have the hierarchical state feedback control model. This synthesizes models of motor control, particularly those that include internal forward models, as well as psycholinguistic models such as the two-stage model. The conceptual system and the word level, like you see in the two-stage model, is not pictured here, but you can imagine it above what's pictured here. And then what the HSFC adds to that are these two sensorimotor feedback loops. And then you would also have external feedback, and you can imagine that below the model. But the point here is to kind of point out how both of these models explicitly include components that relate to auditory sensory information as well as somatosensory information. And then let's get to sensorimotor integration, or impairments in aphasia. So this type of impairment is often implicated in conduction aphasia because of their unique impairment profile. So while fluency and comprehension is relatively preserved, they have a lot of difficulty with repetition. Historically, it's been thought of as a disconnection syndrome, but more recently, area SPT has been implicated. You can see SPT highlighted in the brain on the right. It's a region in the posterior temporal parietal junction, and it's been shown to be active during both phonological short-term memory tasks as well as speech production tasks. And lesions to this area are associated with poor non-word and real word repetition, even after you account for damage to white matter pathways. But what we really don't know still is to what extent are these deficits common across aphasia types. Many aphasia types have difficulty with repetition, but we don't really know much about sensory motor impairment outside of conduction aphasia. And also, how does this impact other tasks? Repetition has been used as a measure of the integrity of the sensory motor system. You have that external auditory stimulus that you have to transform into a motor command to be able to repeat. But how do these impairments manifest or affect other types of language functions? So there were two major aims to the project, and the first was to look at behavioral and neurophysiological correlates of sensory motor impairment in individuals with aphasia using an altered auditory feedback paradigm. And the second major aim was to determine whether compensation in response to altered auditory feedback, or AAF, accounts for any of the variance in performance on language tasks. Particularly here, we'll be talking about a naming task. So there were three major questions associated with the first aim. And the first was, how does speech compensation behavior in individuals with aphasia differ from control? So we're just looking at the behavior in response to the altered feedback. So just to give you an idea of the task, it's been done a couple of different ways, but often participants will be asked to sustain a vowel sound. And then some part of the vowel may be manipulated. Sometimes it's formant structure. In our case, we shifted the pitch either upwards or downwards 100 cents. And that's equivalent to a semitone or the difference between two adjacent keys on a piano. So it is something that you can notice just in everyday life, but they're asked to disregard any changes in their speech and to not try to fix it or do anything else. So previous research has indicated that in neurotypical adults, when they're provided this altered feedback, they try to compensate for this change. It's an online response, and typically they'll shift their voice in the direction opposite of the perceived change. So in this image on the right, it shows some of those responses to AAF. So in part A of that figure, that would have been a response to an upshift because you see the curves going down. And on the right, that would be a response to a downshift because you see the curves going up. So there's very limited to no research examining this in aphasia. This figure is data from a subset of the individuals I'll be talking about today. And as you can see, the individuals with aphasia, their performance is in red, and the control individuals' performance is in gray. And as you can see, it seems that they're compensating to a lesser degree in both directions than the controls. So we weren't sure exactly what to expect. It may be that individuals with aphasia don't compensate to the same degree, but we did expect, because they have a variety of lesion locations and other differences, that they'll demonstrate more variance in the degree of speech compensation than the control participants. So our second question was looking at the ERP components that potentially are markers of impaired sensory motor integration as measured by our AAF task. So again, this has been examined some with neurotypical adults, and compensation in response to AAF has been associated with N1 and P2 components. Normally you see a bilateral and frontal distribution, as you can see in this image here. The scalp distribution, that dark blue is kind of bilaterally distributed. And then in this particular study, they had the participants do a couple of different behavioral tasks, and they correlated the latency of the N1 response with a measure of auditory processing efficiency. So they didn't see that same correlation with the P2 response, so they hypothesized that maybe the N1 is more connected with detection of the error, and the P2 is more correlated with correction of the behavior or the motor side of it. And so our final question of this aim was, how does the reconstructed source of this ERP activity differ between our two groups? So once again, there's a couple of studies that have looked at this with controls, but the ones that have done the source reconstruction typically have used MEG, but many of them use a similar paradigm, and they've uncovered sources in bilateral auditory cortex, temporal regions, supramarginal gyrus, premotor, and primary motor cortex. So with our controls, we did expect to find some of these same sources, particularly auditory cortex associated with the N1 and detection of the modulation, and then motor and premotor sources associated with the later compensation for the modulation. So our second aim, again, was trying to connect this sort of basic function with performance on a higher level language task. So our first question was whether speech compensation in response to AAF correlates with corrective behavior on a naming task. And the idea here was that individuals who demonstrate less speech compensation on the AAF task will probably also be the ones who have more difficulty self-correcting during a naming task. If you have difficulty kind of with an automatic detection of errors, then it may be also more difficult when you have to do that consciously. And then the second question was looking at which regions of neural damage are associated with impaired sensorimotor integration and self-correcting behavior. So if we go back to our models, each of the components of these models have been associated with particular neural regions based on neuroimaging studies. And in both of these models, area SPT is relevant. So for the DIVA model, the posterior perisylvian region, which is highlighted here, you can also see it in the right hemisphere in that image, has been associated with the auditory error map. And then in the HSFC, SPT has been associated with the coordinate transform and that higher level sensory motor loop. And what that component does is facilitate communication between auditory and motor systems. So in both cases, damage to area SPT would likely certainly impair integration of sensory and motor information. So we're expecting that those individuals who do have this pattern of damage will perform poorly on both types of tasks. So I'll go on to how we went about looking at this. We initially set out to recruit 30 individuals with aphasia, as well as 15 age-matched controls. The aphasia group were all individuals who had had a single left hemisphere stroke. They had an aphasia diagnosis as classified by the Western Aphasia Battery, and they were all chronic strokes, so a minimum of 6 months post-stroke. And for both groups, exclusion criteria included a self-reported history of dementia, TBI, psychiatric disorder, or a history of alcohol abuse. So I mentioned before the altered auditory feedback task, and you can see a schematic of that on the left. So again, the participant is cued to sustain a vowel sound. In this case, it was ah, and they hold it for about 2 to 3 seconds. During that time, their pitch is shifted either upwards or downwards, 100 cents, and they're asked to disregard the shift. They repeat this over and over. And there's also listening trials where their voice is played back to them, and again, the pitch is shifted either upwards or downwards. They were also administered a picture naming task, and in this task, they were presented with both pictures of objects as well as abstract pictures, and they were asked to name the pictures of the objects and remain silent for the abstract pictures. And for both of these tasks, speech and EEG signals were recorded. So this is just to remind you of what our aims and questions were, and I'll start by looking at speech compensation behavior. So we ended up including 24 individuals in the final analysis. There were 4 who were unable to complete the task. 1 was excluded due to fatigue, and another 1 was excluded who scored just above the cutoff for the WOB. And you can see some of their demographic information in the table here. They're about 60 years old, about 5 years post-stroke, but there's a wide range there. And the average aphasia quotient on the WOB was about 61. The control group, we had 20 individuals, and they were about 60 years old as well. So for our first question, we extracted the pitch frequencies from the speech signal and then averaged each individual's trials for upward and downward shifts separately. And the speech response magnitude was extracted at each individual's peak latency. So when we averaged these trials, we saw curves like you saw in that figure a little earlier. And so for each individual's peak, we measured the magnitude of that peak. We then conducted a 2-way ANCOVA, looking at group and stimulus direction as main factors and controlling for latency of the speech response. So what we found, these are, if you look in the table, these are all of the response times in milliseconds. And what we found was that for both groups, responses were quicker to the downshift than they were to the upshift. And also we found that in the aphasia group, they tended to respond slower than the individuals in the control group. In terms of the magnitude of the response, there was a larger magnitude response for the downshift in both groups. And in this table here, you have the average response time and standard deviation, but you also have the response time adjusted for differences in timing, and that's highlighted in gray. So interestingly, the aphasia group compensated to a similar degree in both conditions, and that's particularly evident if you look at the adjusted means in gray. And we were also expecting that there would be more variation in performance for the aphasia group, but that actually wasn't true for both conditions. For the downshift, there was actually less variation, whereas for the upshift, there was more variation. But overall, the levels were very similar in both groups. So then we conducted an ANCOVA, and the model ended up being insignificant, so there were no significant main effects after controlling for differences in the timing of the speech response. Okay, so now we'll take a look at the ERP components that correspond with the behavioral compensation. So the sample decreased by 2 for this analysis. After pre-processing, 2 of the individuals with aphasia were excluded because too many of their trials had to be thrown out because of noise. And the same is true for 1 individual in the control group. Our EEG data were pre-processed in EEG lab. The data were re-referenced, filtered, and interpolated. And we also did an ICA to identify and remove any other noise. And then the data were imported into Curry 8 and analyzed. They were epicked, baseline corrected, and then we conducted a topographical analysis of variance, which the name of that is a bit misleading because it's not actually an ANOVA. It's a non-parametric randomization test. And so what is done is the topographic maps are averaged. So you might average across groups or across conditions, depending on what contrast you'd like to look at. And so then the dissimilarity between 2 maps at a given time is calculated, and that's the dependent measure there. So first, just looking at the timing of these components, we found that the latencies were delayed for the aphasia group for both ERP components. So it took a little bit longer in that group to reach the peak latency for each component. These were some of the topographic differences. So this is during the N1 time period at 140 milliseconds. So if you imagine looking straight down at somebody with an EEG cap on, their nose would be at the top, ears at the side, and back of the head at the bottom. And in the top row, we have the controls, and the bottom row we have aphasia. So what we see with the controls is that bilateral frontal distribution, whereas with the aphasia group, we're seeing more diminished activity and increased right lateralized activity, particularly in response to the downshift. In terms of the P2 time period, this is from 230 milliseconds, and we see that same sort of distribution in the controls, that bilateral sort of even distribution, whereas in aphasia, again, it's diminished. And then for the downshift, we see that activity occurring mostly in the left hemisphere and frontal as well as posterior regions. And these are the results for the topographical analysis. So again, we were comparing, at each time point, we were comparing average topographic maps across groups to determine whether the topography of those maps were significantly different. And we found that there were significant differences essentially across the whole time range, so from 29 to 500 milliseconds. And this figure shows the mean global field potential for each group and condition. And the peak occurring between 100 and 200 milliseconds corresponds with average N1 activity for that group. So I don't know if you can see this. Right here, this would be the peak for the controls in response to the upshift. And then the peak occurring between 200 and 300 milliseconds corresponds with the P2 for that group. And you can also see in these curves that the aphasia data is more noisy, and the curves are much smoother for the control individuals. All right, so let's take a look at the ERP activity. So the source reconstruction was also done in Curry. Head models were constructed from T1 scans for the individuals with aphasia. For the controls, we just used standard head models. And then sources were reconstructed. And then we conducted a statistical analysis on the sources to determine statistically significant differences between speaking and listening conditions. So on the right is sort of a flow chart of what we did. So the source analysis requires a lot of time and data and energy, so we wanted to narrow down the time windows that we examined for that analysis. And so first we did a within subject TANOVA. So we were just looking at differences between speaking and listening conditions for an individual. And what we wanted to see was, so we use that contrast in order to isolate sources potentially related to correction of the error, because they would be detected in both conditions. So once those time windows were identified, sources were reconstructed for those time windows, and then we did a statistical analysis for the sources. That analysis is very similar. So you're looking at average sources for speaking versus listening in a single individual, and comparing to see whether the difference in those locations is statistically significant. And then we get the time windows that do have those significant differences. So for the controls, we started out with our 14 participants, and after doing the within subject TANOVA, there were only 13 individuals who showed significant differences between the speaking and listening conditions in terms of the topographic maps. So those significant time windows were entered into, or sources were reconstructed for those windows. And generally, we were looking at between 100 and 300 milliseconds after the onset of the perturbation, and that covered both of our ERPs of interest. So sources were reconstructed for those time windows. The statistical analysis was done on the sources to find significant differences between the conditions. And then we found that only 6 of the 13 showed those significant differences during the N1 time range, and then 5 of the 13 showed those differences for the P2 time range. And those differences are here. So what you have in each row is a different participant. The second column shows you that time window where the significant differences occur. And then you have the rows with one brain, that indicates that the source was consistent for the whole time window. For the rows that have multiple sources, these significant differences change locations within that window. So what we're seeing is that these differences are primarily occurring in temporal and parietal regions of the left hemisphere, including the superior temporal gyrus and inferior parietal lobule. And then we're seeing some later differences from about 180 milliseconds to 200 milliseconds. After the perturbation, we're seeing differences in premotor and motor regions. For most individuals, the significant windows occurred during and after their peak N1. So these results, and in particular those later premotor sources, may be more representative of a transitional period preceding the P2 and that actual motor compensation. And so these are the differences occurring between 200 and 300 milliseconds. And we're seeing that primarily in motor and premotor regions, as well as middle temporal gyrus, inferior temporal gyrus, and some temporoparietal regions. So we then did the same things with the individuals with aphasia. So this time we included the 21 participants. One was excluded who didn't complete the listening portion of the task. And so we conducted the TANOVA. Only 13, again, had significant differences between the topographic maps, between conditions. So for those time windows, sources were reconstructed. Statistical analyses were conducted, and then we found that only 3 of the 13 had significant differences for the N1 and 4 for the P2. So this is very similar to what you just saw. Each row is a different participant, and in the top of the row you'll see the T1 scan, and again the sources, if they're consistent, you'll see one image, and if they change during the time window, you'll see multiple images. So we're really seeing similar sources to controls, although we have much fewer individuals in this group. The sources are often occurring in temporal and parietal regions. However, we are seeing this happen in the right hemisphere more often, which may be expected as a result of the lesions in this group. From 200 to 300 milliseconds, there's a lot of activity in left parietal regions, and for the majority of the individuals, except one, the significant windows occurred after their peak N1 or before their peak N2, similar to the controls, so a transitional period before the motor compensation takes place. So then I tried to sort of connect these results with the behavioral data. So why do some people show significant differences and others do not? There is no clear relationship in the control group, at least at this point. So the individuals who showed those source differences weren't consistently compensating to a greater or lesser degree, and they also weren't compensating earlier or later than the others. The same is true for the aphasia group in terms of the timing and magnitude of the speech response. But there may be some relationship with WOB fluency scores. For the subset of people who did show significant source differences, their fluency scores tended to be higher. And the same is was true for spontaneous speech, part of which is determined by a fluency score. But you can see those means here. I'll move this out of the way. So for the subset, we see that for both of those scores, for both of those sub-scores, it's higher in the control group, although again, this relationship was not necessarily consistent. So just because you did have a higher fluency score doesn't mean that we saw those source differences. All right, so just to summarize what we've done so far, in terms of speech behavior, the individuals with aphasia took longer to compensate, but their compensation magnitude was similar to that of the controls. There was also very little difference between groups in terms of the variability in the response magnitude. In terms of the ERP components, we saw significant differences in activity from essentially across the whole time range, and the latency to the peak was longer for the aphasia group. We see that frontal bilateral distribution of activity in the control group for both components, but for the aphasia group, there was diminished activity overall. In terms of the sources, we found significant source differences in left superior and middle temporal gyrus, inferior parietal lobule, and premotor and primary motor cortex. And we saw a similar distribution of sources for the aphasia group, although they more often occurred in the right hemisphere. Okay, so we'll move on to the second aim by first looking at the naming task and how performance correlated with behavior in response to the AAF. So after the participants completed the naming task, their responses were scored as one of these four. So if they named the item correctly on the first attempt, it was scored as correct. If they initially provided an incorrect attempt and then a correct attempt, it was scored as a successful self-correction. An unsuccessful self-correction was if they were initially incorrect in their attempt and continued making incorrect attempts, or if they provided a correct response and then self-corrected to the wrong response. And then an uncorrected error is just a single response that was incorrect. So from this, we calculated the total amount of self-correction attempts out of the total amount of errors, and we looked at it. We looked at both the totals, the complete total, as well as successful and unsuccessful attempts separately, as well as the proportion of total self-corrections that were successful versus unsuccessful, and then conducted a correlation analysis. So we found that the correlations between speech compensation in response to AAF was non-significant, but we did find that 2 of the 5 measures of self-correction were correlated with lesion size, which would be expected. However, there was no linear correlation between speech compensation and lesion size, and that may be a result of the wide variability in performance. So then we also conducted a region of interest-based lesion symptom mapping analysis. And so we only examined regions that involve the left temporoparietal junction, as well as we included Broca's region as a contrast region. Lesion size was included as a cofactor. And those regions that we included were inferior parietal cortex, supramarginal gyrus, angular gyrus, superior temporal gyrus, and then pars opercularis and pars triangularis. And from this analysis, no region survived, and both the measures of self-correction from the naming task as well as the speech compensation measures were entered as dependent variables. So just to summarize those, there wasn't much in terms of these results. So we had a lot of no results for this aim. So there's no significant correlation between the speech compensation in response to AAF and then the performance on the naming task. And again, the idea was that individuals who have difficulty with error detection that is automatic and unconscious will also have difficulty when those errors are more overt. But I think it's still worth examining further, possibly with a modification of the method or analysis. And then with the lesion symptom mapping, again, no region survived, but it is quite a small sample size for that type of analysis. And in small samples, lesion size plays a big role. So we'll see how that, if those results stay the same with a larger sample. Okay, so what I'd like to do now is try and situate those findings in the context of previous work that's been done, but I'll focus more on the first aim here because there's not much that's been done in terms of either of the major questions. But I'll also suggest some reasons why we may not have found the results we expected and suggest some avenues for building upon this work. So in terms of the speech compensation behavior, we've seen with this and with previous work that given an unexpected alteration in speech output, an online automatic response is generated that typically occurs opposite of the perceived change, and we saw that here. We found that responses were larger to the downshift than to the upshift, and this has also been found in previous studies. And however, we didn't find the same disparity in magnitude, like I showed you in one of those early figures. However, that analysis was done with a smaller set of individuals, and they also didn't control for differences in speech latency. And the variability in the magnitude of the response was comparable across groups, which is quite unusual for aphasia. We're always discussing sources of variability in aphasia, things like lesion size or lesion location and how that impacts performance. But it seems that here those factors play less of a role. In terms of our ERP findings, the N1 and P2 have previously been identified as corresponding with speech compensation to AAF. And again, this has really only been done in typical adults. And the topical distribution that we found here was comparable to previous findings. The aphasia distribution we don't really have anything to compare it to at this point, but it doesn't conflict with what we may expect from individuals who do have MCA damage. But additional studies and larger samples are required to confirm this pattern of activity. In terms of the sources, there's very little previous research, even with controls. But those that have done this have used MEG and have focused more on the N1 component than the P2 component. So Hanks and Maldonado did a study and found sources in bilateral auditory cortex and found that the intensity of those sources was greater for listening than speaking. And Court and colleagues conducted a study in 2014 with a very similar paradigm to what we've done here. However, they selected their regions of interest in advance and looked at peaks at each region over time. The ROIs that they included were the primary auditory cortex, superior and middle temporal gyri, ventral supramarginal gyrus, and posterior superior temporal sulcus, as well as premotor cortex. And they found enhanced activity in all four of those regions during speaking. But they also found overlap of sources for speaking and listening in the ventral SMG and posterior STS, which may explain why we didn't see differences in a number of our participants. And then, as I mentioned before, Korzikov and colleagues looked at the latency of the N1 and were able to correlate it with a behavioral measure of auditory processing and weren't able, and found that there is no correlation with the P2. So they posited that this may instead be, the P2 may be instead related to motor processing. So the control source differences that we found here show overlap with those found in the court study, and the source differences we found in premotor and motor regions also correspond with what Korsakoff posited about behavior associated with the P2. Our aphasia source differences were similar, but again, they more often occurred in the right hemisphere. We don't have anything to compare this to at this point, but this may be expected as a result of their neural damage. And not only that, the aphasia and control sources showed overlap with brain regions identified in those two speech models as critical for error detection and correction. So in DIVA, they identified the posterior STG, anterior SMG, and in the HSFC, STG and STS, anterior SMG as well as SPT, and primary motor cortex were identified. But for the future, it'll be important to make specific individual predictions based on the speech models to get a better idea of the range of typical variation, as well as what happens when these critical regions are damaged. So some questions we still have are, why do many individuals show no difference between conditions? And we may be able to understand that a bit better by looking at additional contrasts. So we contrasted the speaking and listening conditions here, but maybe in the future we could contrast speaking with a perturbation compared to speaking with no perturbation. And then the other question is, what's the relationship to the behavior? It wasn't clear if those with why those with differences didn't show greater magnitude compensation or quicker responses, but teasing that out will be important later as well. So there were a number of limitations that we faced while conducting this portion of the study. One is the TANOVA analysis method, which was new to me and I think to a number of us. But because the trials were averaged across, average within an individual, we're really comparing differences in brain activity and not necessarily behavior. So an individual is going to vary in the magnitude and timing of their response, and we didn't identify individually define trials as either low compensation or high compensation. However, this relationship between the N1 and P2 components with the speech compensation in response to AAF has been documented multiple times before, so I think it's safe to make that assumption for now. Another challenge was examining ERPs and reconstructing sources in damaged brains. So our ERP data was much noisier for the individuals with aphasia, which made it difficult to retain enough trials for analysis and required a lot of trial and error in terms of pre-processing. And also the variation in lesion location didn't allow for a group analysis of the source locations. So because there is so much variation in those sources and variation in lesion site and size, it's difficult to generalize this to a larger population. The sample was a particular limitation for the lesion symptom mapping analysis. Again, lesion size drives findings with small samples, and the distribution of aphasia types was unequal, which may cause some bias in terms of lesion location. And in terms of the naming task and scoring mechanism, it was a novel scoring mechanism that was used, and it may not be sensitive enough. And the task included 80 pictures of objects and 40 abstract pictures. So it could be that the task wasn't long enough to get enough samples to detect these subtle relationships. So because this study has really just scratched the surface with this topic. There's a lot of room for future direction, so I just make a few suggestions here. I think it would be interesting to group trials by response magnitude for ERP analysis. So how does the topographic distribution of activity change for, or look different for low compensation or no compensation trials versus full compensation? Or what are the differences in timing? Also looking at the degree to which the shift is detected overtly. So they're asked to disregard the shift, but it's unclear how many participants are actually overtly noticing the shift and how many are not. And it would be interesting to see how well that corresponds with behavior. So are the individuals who are detecting the shift consistently, are they compensating consistently as well? It will also be important to group individuals by aphasia type, which would require a larger sample, but that can hopefully allow us to do some group analyses when it comes to the source analysis. And then also, it would be interesting to look and see if compensation behavior can be modified, either through behavioral treatment, explicitly or implicitly, and it would also be interesting to explore the effects of neurostimulation. So to conclude, there are potential markers of sensorimotor integration, both behavioral and neurophysiological markers. We successfully replicated the behavioral compensation and topographic patterns identified previously with controls, and we extended this to a clinical population. It serves as a first step towards understanding sensorimotor impairment across aphasia types and markers of that function. However, we still have a ways to go in terms of understanding the relationship with higher level language abilities. So, thank you, and I'm happy to take any questions you have.
Okay, so what we typically do is you take questions from the room first, and then we'll see if there's online questions as well.
Okay.
Oh, and if you get a question from the back of the room, please repeat the questions.
Okay. Yes.
Question from the back of the room. So you mentioned that people who have aphasia, they're basically slower to correct for the altered audio feedback, right? And in terms of their behavioral response, and you also see this delay in terms of their ERP response, right? Does that delay correlate with fluency or with WOB IQ?
The delay in the ERP, well, so the question was, does the delay in ERP response
As well as behavior?
As well as behavior correlate with WOB IQ?
Or fluency?
Or fluency. So that's not something I've looked at yet, but I think it's an interesting question. Mm-hmm. And I think very relevant too, so I will do that and get back to you. Yeah.
So my understanding in the literature, based on the control side of this equation, is that the shift that you do in response to an upshift is pretty much of equal magnitude back. So it's as if you're shifting down. Did you see that in your controls? And do you have a sense from the data that the aphasics, are they doing something more that they're missing the target of the shift, or are they sort of undershooting or overshooting in a systematic way? So is it more, because you say, because the interesting was it was less variable, it seemed like, so I'm just kind of wondering how that data plays out there.
Yeah, so the question was, you mentioned that in previous literature they show the compensation is similar to the degree of the shift, and kind of my thoughts on why the individuals with aphasia may be compensating less, or what are they just missing the mark? So from what I've looked at, the compensation actually seems to be less than the actual magnitude of the shift, but I don't have a great response for the aphasia piece. It's not clear whether it just takes longer to, and it seems like from the data that it just takes longer, and given an extra couple of milliseconds, they'll get to the same compensation. But it doesn't seem like they're just completely missing the mark. And part of the issue in kind of making those conclusions was that there was so much variability. So there were people who were overshooting every single, well, in the aggregate when the trials were averaged. And we had a couple of people in each group who followed the shift. So instead of compensating opposite, they went the same way as the shift. So it's hard to say at this point, but my guess would be it's more, rather than missing the mark, it's more needing time to get to the mark.
So it seems to me that that focus on the individual differences between those folks is really the key, right? And
yeah.
One could even wonder whether it actually makes sense to do this as a group analysis, given that you would already assume, I would assume, that the speakers with aphasia that you had in your sample all have widely differing problems, right? So it seems from your own data, if you aggregate the data, your real conclusion should be there's no trouble with sensorimotor integration in aphasia. And, right? Because you don't see big differences between the
In terms of the
aphasia.
magnitude, sure, yeah.
Right, but you obviously think that there is. So what types of aphasia you think, or what particular language disorders do you think would show up as resulting in different results on the compensatory behavior? Just behavior.
So that's an interesting question. One thing I would say is I think it was a good choice to just look at them in aggregate as a first step. And compare that to what we've seen previously in controls. In terms of what sorts of disorders I think may show, so the question of what sorts of disorders may have difficulty or show decreased compensation. I think based on what we know about conduction aphasia, that would be the obvious choice. But I'm not so sure when it comes to other aphasia types. I definitely noticed those with apraxic speech had more difficulty with the task. However, they had more difficulty in terms of fatigue and being able to withstand the whole length of the task. They were doing this for about 60 minutes.
And they might have trouble with maintaining the tone in the first place,
Yeah,
right?
exactly.
And regardless of feedback.
Right, and I was just going to mention that I did look at some of those individually, and the response was, it wasn't a diminished response necessarily. I think it's hard to say though, exactly, just based on the expectations for these data and them not coming out exactly as we planned. I think it's hard to say just based on behavior at this point who may be more likely, just because that relationship isn't clear yet. Yes.
So, do you have any insight or thoughts on the pre-processing and the final processing of the EEG data in people with lesions? It seems to me, I know you've had a lot of experience with that now, and I'm sure that a lot of people would like to hear about that.
Yeah, so I think the best thing to do happens before the pre-processing, and that's getting as much trials as you can. Stacy and I started with recording about an hour of data, and after we started pre-processing and seeing that a lot of trials were being rejected, we started recording as much as the participant could withstand in a lot of cases. So I think getting as much trials as you can beforehand is important, because just averaging them will get rid of some of that noise. And there's a lot of parameters you can sort of play with in terms of the pre-processing pipeline. So rather than just selecting, and it's more time consuming to pre-process individually, but rather than selecting parameters to reject trials for eye blinks that are the same across individuals, maybe people have smaller or larger magnitude eye blinks, so more of these trials are rejected, and then you're rejecting those trials in others who don't have as large of a magnitude in that case. So I think you just have to get as many trials as you can in the first place, and then play with the pre-processing parameters for your sample, because it'll vary.
What about the lesion and its effect in the source analysis?
That's a big question. So I think these source analyses really have to be taken very cautiously. Source reconstruction is a difficult process in itself, even for people with non-damaged brains. So some of the things we did was just not aggregating the source results because everyone has different lesions. That's going to affect propagation of the electrical activity to the scalp. But one thing that I think is very helpful is having individual MRI scans for all of them, because all of their head models were based on their scan, and that would probably give us much better results than if we use a standard model. But I think there's a lot of considerations that have to be taken. So if you do want to do a group analysis, use individuals with similar lesion sizes and locations and controlling for those factors ahead of time. Yes?
So most of the models that are there, they have a bilateral distribution such that both hemispheres seem to be participating in this. And I don't know the literature on this at all in compensation. If you were presenting people with these on just the left or just the right hemisphere, would you expect to see differences, especially among the phasics that have lesions to the left hemisphere? And would that be a way to potentially move forward on this kind of data?
That's a great question. So the question was, well, first stating that we see bilateral distribution of these sources, and if we would see differences in source activity if we present the sound or the altered feedback to the left ear versus the right ear. I'm not sure about the answer to that one, but I would expect that we might see similar results to what we have now because they are presented bilaterally. I'm not very sure. I'll have to think about that one a bit more, but I think that's a great point. Any other questions?
We have no online questions, so you're done. Thank you very much.